All right, wonderful. Thank you so much. Justin, I really appreciate the introduction. Azure, I appreciate the invite. And I just want to say briefly to everyone before I get started, I'm a big fan of Azure Systems. So I was kind of an old Lycor hand back in the day before Azure was around. And so you find a piece of equipment that works for you, you tend to stick with it. But I've been working with Azure for a couple years now, and really I'm a big believer in their system. We have one in our lab, obviously. It's produced great data. These are great people to work with. Really, just it's been a wonderful experience working with them and working with their equipment. So I highly recommend, and happy to answer any questions, or if you're in the area, even you can demo the equipment, whatever you want. This is a good group of people. I'm happy to be a part of them. Happy to be a part of this. And I'm happy to be here today to tell you about some of our work. So I'm very interested in pain and chronic pain. And so that is taken a lot of different forms in our lab. So opioids would be one big area of research. Also, cannabinoids and cannabis have been another big area of research. And recently, we've gotten interested in these compounds called terpenes. We'll go into a bit more on exactly what terpenes are. But these are sort of a different component of the plant that many people have been looking into. And ultimately, we are very interested in treating chronic pain. But of course, the whole concept of cannabinoids, cannabis, medical cannabis, and so on is much larger than pain and much larger than chronic pain. And so that's part of the reason why understanding this plant and its constituents is important. And of course, one would be the fact that this, there's, beyond scientific, beyond even medical, there are social components to this as well, political and so on. And so cannabis is until recently was a legal drug. You couldn't use it for any purpose, including medical. I can remember those days when it was fully illegal. And, people use it for recreational purposes, as well as for medical. And one reason to study this, as you can see by this sort of graph of drugs over the years, is many illegal drugs have sort of stayed flat and have not increased in their use, but cannabis has actually gone up. So more people are using cannabis, and that's not necessarily a bad thing, right? So there are legitimate purposes that you might want to use cannabis. And to be clear, I think recreational use is also legitimate as long as it doesn't become destructive. But as this is increasing, so it behooves us to understand what's going on. And in addition to that, I should turn on my laser pointer instead of this little guy. And in addition to sort of that general increase in use, youths have increased their use of marijuana as well, whereas for other drugs it's stayed flat or even gone down. So we really should understand what's going on with this plant and its constituents. But in general, I will say we don't really understand all that well what's going on. And so, of course, the real meat and the real impetus behind all of this has been the change in legal status of marijuana. And so many states have now made it legal for recreational and medicinal use. Other states have made it legal for medical use. And then, of course, there are some where it's legal for neither. It still remains a fully illegal drug. And that legalization, that change in status has really, been fueled part of this boom and this increase in use. And it's more than just that, right? So when something becomes fully legal, now it's legitimate for companies to get into this market, to create and new products, to be able to advertise, to sell. And so along with this boom in use has been sort of what I would almost term a gold rush of commercial activity, of all these companies that are jumping in. Of course, many of them are making claims that are just scientifically unsupported, right? So this, use cannabis, it's going to make your life better in 1,000 different ways. It's going to cure this disease. It's going to cure that disease. And of course, there's really no evidence behind many of these claims. There are behind some, but behind many of these claims. And so from a scientific point of view, from a medical point of view, we also need to understand how this plant works and what's going on so that we can bring some order and some scientific rigor into this commercial space that's rapidly leaving us behind. And so what is cannabis? It's a plant. I think you all know that. Shown here is the flower or the bud that has most of the active constituents present. Even on that bud, there's these little structures called trichromes that have the heaviest concentrations of active component. The active component for our cases being cannabinoids and terpenes. So that's really zoomed in here. You've got these little structures. This has most of the business end of the plant, but most of the chemicals. And there are two main classes of drug or of chemicals that we're mainly concerned with. First are the phytocannabinoids. So you have everyone's favorite delta-9 tetrahydrocannabinolic acid, also known as THC. So this is sort of the main psychoactive cannabinoid present in the highest concentration that people think about. There are also other psychoactive cannabinoids present, but they are present in lesser concentrations. And also these are very poorly studied. So people have studied THC, of course, but many of these minor cannabinoids have barely been studied at all, if at all. And so that's a big and up and coming area of research that needs to be looked into. And in addition, there are these terpene compounds. So terpene compounds are simpler chemically. I'll show you in a moment what that looks like. They provide aroma and flavor to the plant, but they also have pharmacological features as well, which we'll get into. And this is just listing some of them. There are more than 140 different terpene compounds which have been identified in cannabis and other plants. So there's a lot of these chemicals. And so one reason you might think that the cannabinoids and the terpenes are going to sort of both have pharmacological activity or share features is that they actually share the same biosynthetic pathway, at least to begin with. So you have these precursor molecules, including glucose, that are converted by these enzymes, and eventually you get to this common precursor, geranyl diphosphate, and this is a bifurcation point. So the geranyl diphosphate can be either acted upon by CBGA synthase to be turned into a cannabinoid precursor to all cannabinoids, or it can be converted by one of two terpene synthase molecules to become monoterpenes or sesquiterpenes. And so these compounds are, share that sort of that common ancestry and that common biosynthesis. So again, a reason to think that they might have something in common beyond that. Sorry, there we go. And so this is a little bit more on terpenes. As I mentioned, very large and diverse class of organic compounds with over 140 identified in cannabis sativa, aroma and flavor. And then you may have heard of essential oils. So, I mean, people, again, have made a lot of bullshit claims, if I may use a little French, a little colorful language. Have made a lot of bullshit claims about what essential oils can do, but they are heavily composed of terpenes, majority composed of terpenes. It's a lot. That's most of the constituent of these essential oils. And various studies have shown that these have impacts. These have pharmacological impacts. So it's not all nonsense. There's a lot of nonsense, but there's also some science that we need to figure out what's going on. And we'll be able to use these for our purposes. So this is listing some common monoterpenes and sesquiterpenes, also shown that they're not just in, these compounds are not just in cannabis. So they're in many different types of plants. Oranges, lemons, limonene and lemon, hence the name, hops, pine. So they're not exclusive to cannabinoids. And some of these studies, sort of briefly listed here, have found a variety of different effects, including anti-inflammatory, anti-anxiety, and analgesic, pain-relieving properties as well, suggesting that they could indeed have some beneficial therapeutic properties. And just to sort of show you the other side of things, so we have the terpenes, we also have the cannabinoids, and they both may be working together. This is that precursor molecule, CBGA, that's created through that common synthetic pathway. And then through these various synthases, this various family of synthases, you have these different cannabinoids and minor cannabinoids that are created. And of course, the one in the middle right here, THC, is the most, the highest quantity and the most studied and sort of most appreciated of the cannabinoids. And in fact, these compounds may be working together. And that's something we'll talk more about in a second. And so how do the cannabinoids work in your body? So they work through this endocannabinoid system. So this is not a system that, of course, has evolved to respond to cannabis. This is a system that has evolved to respond to endogenous signals in your own body. These endogenous ligands, mostly lipid-based, including anandamide and 2-arachidonyl, also known as AEA, 2-arachidonyl glycerol, I chopped that up to begin with, 2-AG, as well as other putative ligands of rhodamine and others. So this system has evolved to respond to these endogenous modulators. And there are receptors through which these signals work and through which cannabinoids, once they've been ingested into your body, work. The highest expression in the brain is this cannabinoid 1 receptor, mostly present on, sorry, this is being really slow for some reason. Mostly present on presynaptic terminals. It is the most highly expressed receptor system in your brain. So it's not like this is a minor thing. There's extremely high levels of CB1 receptor in your brain, the highest expressing modulatory receptor system there is, expressed in nearly any circuit you might imagine has cannabinoid receptor 1 modulator. So this is important. CB2 receptor present mostly in the periphery and is heavily involved in regulated inflammation, being looked at for inflammatory pain, as well as some others that have been identified. GPR55 seems to be pretty solidly a cannabinoid receptor. There may be others. And so we have this kind of this important modulatory system present all over your body that does lots of things. And this is what the cannabinoids are acting through. And then on the back end, you also have enzymes for synthesis and breakdown. So all living systems have negative feedback loops associated with them. You have these enzymes that break down the signals, the endogenous ligands, and terminate the signal. And these enzymes are also targets for study. So you have enzymes like FA, fatty acid, something hydrolase, MAG lipase. So these enzymes are being targeted through therapeutic means to increase the levels of endogenous cannabinoid to treat pain and other conditions. Some of these have even gone into clinical trials. So these are major targets of study as well. So you have this important system through which these plants are working, and keep that in mind as we study these things. This is sort of the system in your body that these will be working through. Oh, by the way, I didn't mention at the start, if anyone has any questions, feel free to jump in and stop me, and we can make this more discussion oriented. You don't have to wait to the end if you don't want to. Sorry. And then, so you have this cannabinoid system, and then there also is some complexity to take into account when you study the plant. So most people are taking this in as through the plant. Either they're smoking it, vaping concentrate, through inhalation, eating extract, orally consuming extracts. Those extracts could be through oil, could be through CO2. So most people are taking some variety of the plant. And so you have this plant, and this plant, it does not just have one component like THC. It has numerous components, right? So we have up to 140 different terpenes, first of all, depending upon which strain of cannabis you're talking about. You have THC, sure, but you have these other cannabinoids that are present as well in different mixes and concentrations. So you have this great complexity of chemical constituents to the plant. And then even the dose is a question, right? So you have the dose is really not that well defined in the ways that many people are taking this plant. So you're smoking, consuming an extract, whatever, that is inherently variable from day to day. You take a dose on one day, you take the dose the next day. That's inherently variable in terms of what you're getting. It's variable between people. So different people are going to be effectively getting different doses. This is not something that's standardized like in a pill. And that makes things complicated and difficult to understand. And that sometimes can leave it difficult to understand how you need to give the drug to achieve your therapeutic effect versus your side effects. What's the best optimal use? And of course, this even leaves aside the whole issue of recreational use. And now let's further add some complexity to this. There's not just one cannabis plant. There are many different strains that people have developed. You've probably heard of some of them. Charlotte's Web being a famous one that's heavy in CBD for the use of a poor little girl, I think it was, that had Dravet syndrome, that had these continuous seizures that this extract helped her with. And so you have these different strains, and each of these strains can have different blends of terpenes and cannabinoids, and then you have the further issues with the dosing, adding further complexity, make it further even more difficult to understand what's going on in terms of therapeutics and treating people. And so one sort of indication with this, or one early sort of idea with this, were these two strains generally referred to as sativa and indica. Of course, it's way more complicated than that, but the basic idea behind this at the time is that you have these two different strains. They have different levels of THC versus CBD, and that led to some different strains that had different blends of THC versus CBD to THC versus CBD. And the thought was that this would give you a different sort of outcome, right? A different therapeutic outcome. And adding terpenes into the mix, this has now been started to look at for terpenes by companies. So this is not a scientific study, unfortunately. This is a company, Leafly, I think it's called, that's started to analyze terpene content in these different strains, started to sort of quantify this. They claim that the different blends of terpene are more effective for different sort of effects. Of course, this kind of looks like bullshit just to look at it to begin with. Inspiration, how do you measure inspiration in the lab? How do you quantify inspiration? How do you say that one drug is causing inspiration versus another? But it at least shows that people are starting to consider the different chemical constituents of these different strains of plant and how they might give you a different effect. And they started to quantify different levels of terpene in these different strains. And this leads us, so all these ideas, these complexity, the different blends of terpene versus cannabinoid, the different strains of cannabinoid, all this variability leads us to the idea of the entourage effect. That's what all this has sort of been leading up to. And the entourage effect is the idea that when you smoke or ingest or whatever cannabis, you're not just getting the effect of THC, right? So you're not just getting one therapeutic or benefit or even side effect profile from this one molecule. You're getting this big, complicated blend, and all these constituents are impacting each other. So you're not just getting the THC effect alone in isolation, you're getting THC modulated by all these other cannabinoids that are present in the plant, such as CBD, CBN, and CBG, and so on. So these are modulating the impact. And then that's further being modulated by the different blends of terpenes that you might have in the plant, and flavonoids and other components potentially as well. So you have this great complexity that's all impacting each other. And as you get different strains and chemovars of cannabis, you're going to get a different entourage effect. This is the hypothesis. And then, of course, the ultimate promise of this hypothesis is that you can use this to your advantage. You can sort of dial in just that right blend of different components that will give you maximum pain relief or whatever it is that you're looking for, minimal side effects, decreasing the psychoactive side effects, and so on, to really give you that dialed in, correct, therapeutic profile. That's the goal. That's the hypothesis. Now, there's unfortunately not a lot of evidence to support that. And this is part of where we're coming in as a lab to get into that. And so again, companies are jumping on this idea of entourage effect, sometimes in kind of a bullshit way. So you have this company Leafly that's now putting out this information. So this is from their website. You have these different terpenes like humulene, pine, and linalool, and some of this is absolutely factual, right? Like the boiling point, the sort of the sensory experience. So that is true. You can't really debate that. But then they're starting to promote it with these different therapeutic effects. So, if treating insomnia, stress, depression, sedating, and calming. So there's no real solid evidence behind this. So that's where we need to come in as scientists and provide some rigor, test this hypothesis, this entourage effect hypothesis, and provide some rigor to this field, and also be able to figure out if you can really use this to our advantage to treat chronic pain, which would be my ultimate goal. And so this is just sort of schematicizing, if that's even a word, this idea that we're going to, as a lab, we want to study entourage effect. We want to look at differential regulation of cannabinoid agonist-induced behaviors, how all these interact with each other. Also, we want to look at the impacts of terpenes and cannabinoids alone, but also in combination with each other. We need to do this in a rigorous way. There is not much evidence in favor of the entourage hypothesis, so it's sort of become popular. It's become social and sort of one of these meme kind of things, but there's not a lot of good evidence for it. So there are a few different studies suggesting that full spectrum extracts or CBD heavy extracts are more efficacious than like THC alone or THC heavy extracts. But again, is that entourage, or just because these are complicated blends, complicated extracts? Maybe there's something else about them that's achieving this benefit. So these are not really good evidence. There's a few negative papers. So there's these two papers in here. This one actually hasn't been published yet. I picked this one up on bioRxiv. This one is published. Trying to suggest or finding negative evidence for entourage, but there's some problems with these. So this study used very low concentrations of terpene, and this study looked at very specific cell outputs in an in vitro model. So maybe it just is not, synergistic in that particular output, but it could be in some other output. So the evidence is weak either way, and that's where we need to come in and do a better job of testing this and figure out what's going on. So this leaves us then with our formal research question: do terpenes interact directly or indirectly with cannabinoid receptors or agonists to modify function and behavior? So how are we going to do this in our labs? What are we going to do in our study? So first of all, these are the terpenes we chose to study. We have beta-pinene, linalool, geraniol, and alpha-humulene. These are sort of common terpenes, not only found in cannabis, so they are found in these other plants as well. Most of them are found at pretty high levels in the cannabis plant. And there are some studies out there, again, not great studies, not high quality studies, but some studies suggesting that they could be involved in these various processes, including potential antinociceptive analgesic properties, which is something we want to study. We also included a control terpene. So beta-caryophyllene, kind of famously a few years ago, a paper came out suggesting that this was a cannabinoid receptor 2 agonist, which could be good in inflammatory pain. So we included this as sort of a control to say, is this CB2 selective, agonist, if you will, or terpene? Is that, is that showing the same effects as these other terpenes? Sorry, whoever could mute John Shallcross, could you please mute yourself? Thank you. And so we have a few different ways we want to study the terpenes. So sort of just schematically here, we have, you can think about this, you can have this is showing sort of THC or any cannabinoid really having some sort of direct impact on the cannabinoid 1 receptor. This could be studied in vitro model. You also have these cannabinoids producing some sort of in vivo effect, behavioral or otherwise, and what that might be. And you have different outputs for how you can measure these in vitro and in vivo. So in vitro, you have outputs like ERK MAP kinase phosphorylation. This is what we used the Azure system for, which I'll get into more detail in a bit. You have other downstream signaling components. In the mice, in particular, we want to study their behaviors, their cannabinoid-like, pain-relieving-like behaviors. And then you have the question of how are terpenes either producing this on their own or modulating the effect of cannabinoid. Are they increasing it? Are they decreasing it? We can look at this in vitro and we can look at this in vivo. And then, of course, we have the question of what mechanisms are the terpenes working through to accomplish these changes in vitro and in vivo. And that's something we'll get into as well. So what we're going to do is we're going to start with the in vivo. We're going to start with the animal model and see what that shows us. And we're going to start with, we just use wild type CD1 mice, nothing particularly special about these animals. We use males and females too. We wanted to look at potential sex differences. And we started with the suite of behaviors called the cannabinoid tetrad. So these are 4 behaviors characteristically produced by cannabinoid treatment. They don't only happen through cannabinoid treatment, but they are produced by cannabinoid treatment. And there are 4 of them, hence the name. First of all, you have hypolocomotion. They decrease their movement, their free movement. And this is shown here. This is not our data. This is from a publication. THC reduces, reversed by Rimonabant, the CB1 selective antagonist. Cannabinoids produce hypothermia, a decrease in core temperature, again, reversed by the antagonist. They have this cataleptic behavior, catalepsy or freezing behavior, measured by the ring test. And so a normal animal doesn't really have this cataleptic behavior at all. And then when you treat with the cannabinoid, you get the catalepsy, it's reversed by the antagonist. And then lastly, and most importantly, or at least most importantly for me, for our studies, they produce an antinociceptive or analgesic behavior. And this can be shown in a variety of tests, tail flick, hot plate, and others. Here, this is the, I believe this is a hot plate test. So they have an increased latency. So in other words, they're leaving their paw in the heat stimulus for longer because they're having an analgesic effect, and that's reversed by the antagonist, the Rimonabant. So keep these 4 characteristic behaviors in mind. And this is what we're going to use to test first in our animals. And so we start with sort of what's most interesting to me, the antinociception, the pain relieving component. And we did perform this using a tail flick test. So just immerse the animal's tail in 52 degrees Celsius water, measure how long it takes them to flick it out. It's very simple. And so a typical animal at 52C has a baseline of around 2 to 3 seconds. And then as they get an antinociceptive effect, they leave their tails in longer. And as you can see, looking at our 4 main terpene compounds, all 4 terpenes produce modest, mostly dose-dependent antinociception, particularly obvious for alpha-humulene and geraniol. So you have this dose-dependent antinociception that's produced, suggesting that they are relieving pain. Beta-pinene and linalool have a little bit of an effect, but not nearly as strong as the other 2. So now we're starting to see a bit of differentiation between the 4 terpene compounds, but they do all produce some antinociception. The beta-caryophyllene, positive control, or actually negative control in this case, produces just the slightest uptick, probably not really any antinociception at all. And this is at a dose twice as high as what was reported in the literature to produce effects at the CB2 receptor. And then we have our control agonist. So keep this one in mind. We use it a lot in this study. We have this WIN552112 or 212, which is a non-selective CB1, CB2 agonist, sort of one of these prototypical cannabinoids, which also produces stronger dose-dependent antinociception in this assay. We're going to compare a lot of our stuff to WIN compound. So first leg of the tetrad's a hit. All 4 terpenes are producing this antinociception, which is good, which suggests that we could use these terpenes for pain-relieving treatment. And so I do want to briefly show our experimental design as we examine the rest of the tetrad behaviors. So we're able to examine all 4 behaviors in one mouse, which, you get that within subjects design, improving your rigor. So we have the male and female CD1 mice. We take our baseline tests before we start treatment. At 0 minute, we inject either 200 mg per kg terpene or 5.6 mg per kg WIN55212 control. And then we have our different tests here at different time points, open field, ring, and temperature, to get the rest of the tetrad sort of in one go with one animal. And so first, let's look at hypothermia. All four of our tetrads, all four of our tetrads, all four of our terpenes, as well as the Win55212 positive control, all produce a robust hyperthermic response. So now we've got two legs on our tetrad, sort of two cannabinoid or cannabinoid-like or cannabimimetic behaviors from terpene treatment, producing this very effectively. Importantly, vehicle control, no hypothermia, as well as the beta-carophylline CB2 selective agonist, no hypothermia, suggesting that this is kind of selective. This is not just a general effect that we see, and that in fact our terpenes are producing sort of this cannabinoid-like behavior. This is further supported by the catalepsy findings. So again, vehicle and beta-carophylline control, no catalepsy, but Win and all four terpenes produce a robust catalepsy, although it varies a bit between the different terpenes, suggesting again that all four terpenes are cannabimimetic, producing these tetrad behaviors. And lastly, we looked at hypolocomotion. So yes, and I include this data because it's instructive. So again, familiarly with the last set of experiments, we do see a hypolocomotion with the Win and the four terpenes. However, we also see it in the beta-carophylline control, and we also see it with vehicle, right? So this should really be our negative. And we saw it with two aspects of hypolocomotion, the distance traveled measurement, as well as the mobile time, the amount spent mobile. All had significant decreases for all treatments. And what we ultimately chalked this up to was the fact that the animals were acclimating. So we remember we have a baseline measurement. That's sort of shown here as the before in every measurement. We have that baseline measurement, so the animals were getting acclimated to the environment. So we put them back into that same environment. They're already bored. They don't do that same exploratory behavior, so they're going to have an apparent decrease in mobile time that's not and distance traveled that really doesn't have anything to do with drug treatment. It's just because they're habituated. So to get around that, we did another set of experiments where we did not habituate, we did not do any baseline measurements. We just straight up injected them and put them right into that environment. And here now we start to see differentiation. So we don't see any apparent suppression with vehicle and beta-carophylline in either measurement, whereas the Win55212 and the terpenes do show significant decreases. Alpha-humulene, very close, not statistically significant, but very close. I kind of chalk this one up too as a decrease in hypolocomotive activity. And so this really now fits with the rest of our tetrad experiments. We now have the fourth leg of the tetrad providing evidence that these terpenes are producing cannabinoid-like behaviors, really suggesting first that these terpenes do have some sort of pharmacological effect in vivo. It's not just an impact of they're not just producing nice smells and tastes. They really do have a pharmacological effect. It is cannabinoid-like, suggesting you could potentially use these as a therapeutic agent for pain relief, and suggesting that they could have an entourage effect, that they could be combining with cannabinoid or other molecules in vitro and in vivo to produce a different effect. And so that is basically what I just said. Our first set of conclusions. Terpenes can induce these behaviors to different extents. Our control compounds did not. And that leaves us with the question, what are the mechanisms of how these are acting? Do they work through cannabinoid or other receptors? And do they interact with cannabinoid agonists to influence activity? An entourage effect, in other words. And so to test that, we have a similar but slightly different experimental design. Again, we test one animal in all behaviors, which is a great advantage of this approach. We do our baselines, and then at minus 10 minutes, we inject one of two antagonists. Rimonabant is a cannabinoid receptor 1 selective antagonist. Istradifilin is an adenosine 2A selective antagonist, which is sort of this similar but not identical modulatory receptor system. At 0 minute, we inject either terpene, either alone or with combination with Win compound, and then we're able to get our 4 different behaviors over this time course and pick them all out in one set. So what does that look like? So first of all, let's start with tail flick. It's by far the simplest. Straight up, we found that tail flick latency increases our cannabinoid 1 receptor mediated. That was sort of very simple in our hands. So first of all, here's our Win55212 control. You see the antinociception. It's fully blocked by Rimonabant. Okay, that makes sense. We then look at all of our terpenes. So this is terpene alone. You see the tail flick antinociception produced to varying extents by all 4 terpenes. All 4 terpenes, you see a complete loss of any antinociceptive activity with the CB1 antagonist, strongly suggesting that all terpenes are producing this tail flick effect through the cannabinoid receptor 1. So same as THC, same as Win, same as anything else you might imagine. So this really, again, provides support for the idea that these are true pharmacological agents that are working through this receptor system. And then we looked at additivity when you combine terpene with the Win compound. So normally the Win compound alone is right about this level, terpene is right about this level. You combine them together, in all 4 cases, we saw additive effects. So we saw greater antinociception, greater pain relief when you combine these 2 drugs than when you have either of them alone. And this provides some support to the entourage effect hypothesis, suggests that these drugs could be combining when you consume them together, and suggests that we could use this therapeutically to combine terpene and cannabinoid at least, or maybe other pain relieving agents as well, to make pain relief more effective. But then you may say, wait a minute, you're making pain relief more effective, but what about the other tetrad behaviors? It doesn't really help you if your therapeutic benefit is better, but your side effects are just increased to the same extent. So we looked at that and we found some differences. And so first of all, we looked at catalepsy. So in each graph, each graph is a different terpene. Each bar graph is the terpene alone. So each terpene alone produces some catalepsy, yes, but it becomes a different story when you combine them with Win55212. So alpha-humulene and beta-pinene, yes, they did synergize in catalepsy with Win55. You saw greater catalepsy than with either alone. However, that was not the case for geraniol and linalool. In both cases, we saw no significant increase with Win as combined. So now you say, wait a minute, so we're synergizing with the pain relief, but we're not synergizing with at least some of the side effects. So again, you might be able to make that right profile to sort of select the right terpenes that will synergize with your pain relief, not synergize with your side effects, to give you an overall better therapeutic benefit. Sorry, I'm blocking a bit of my screen. Move that. Okay, and then so that's synergy or combination with the Win compound. What about mechanism? So as I mentioned just previously, we found CB1 could explain all of the tail flick, but we could not find that same explanation when we treated with Rimonabant and catalepsy. We saw a partial reversal with Rimonabant treatment in alpha-humulene and a partial reversal with linalool. So CB1 may be somewhat involved in these 2 terpenes, but not nearly to the extent that it was in tail flick antinociception. And we saw no impact of Rimonabant at all on geraniol and beta-pinene. No significant reduction of that effect, suggesting that CB1 is not involved in this tetrad behavior with these particular terpenes at all. And just to remind you, so we have our Win55212 positive control. We did see a very significant reversal with Rimonabant treatment with the Win. So it's not just that you can't reverse this behavior, you can. And so this now gets into the idea that the terpenes are not just acting through one target, that they are a polypharmacological drug. That yes, they're acting through CB1, but they could be acting through other receptors and mechanisms as well to produce relevant behaviors in the animal. And this was supported by our istradifilene experiment. So remember, this is an antagonist for the adenosine 2A receptor, different from the CB1. And here we saw a very strong effect, full reversal of catalepsy in alpha-humulene and beta-pinene, suggesting that this receptor system has a strong role for these terpenes in this behavior. So cannabidiol, but not going through CB1, going through the adenosine 2A to achieve these behaviors. And then we saw a partial reversal with istradifilene for the other 2, geraniol and linalool, suggesting at least a modest role for this receptor system. So now we've identified at least 2 targets that these terpenes are working through, cannabinoid receptor 1 and adenosine 2A in vivo, to produce these effects. So these are important mechanistic insights. And we found a different story yet when we looked at hypothermia. So hypothermia, we looked at, so again, you have each drug alone produces the hypothermic effect. Now, in this case, a bit different from the data I just showed you, we did see at least some synergy with Win in each one, right? So now we do have an entourage effect for the hypothermia. However, when we looked at Rimonabant, we saw a partial reversal with alpha-humulene, but none of the others. So CB1 is only seen to be mildly involved with alpha-humulene, but none of the others. And as well with adenosine 2A, we saw such a strong effect in the catalepsy, but only a mild reversal for alpha-humulene, no impact in the other behaviors. So not only do we have multiple receptor systems, but they vary, which one is being utilized, which one is being targeted varies for each terpene depending upon the behavior that we're talking about. So we know that CB1 is involved, we know that A2A is involved, but they're involved to different extents and different behaviors, and we know that there are other receptor systems present that are not explained by these results. So these terpenes are potentially doing a lot of things. And then lastly, we looked at hypolocomotion, the last leg of the tetrad. And again, we saw some variability. So we saw a little bit of synergy with WT 55212 and at least some of these measures for beta-pinene, alpha-humulene, and linalool. Did not see the synergy with geraniol. And we didn't really see any impact at all of Rimonabant. We saw no CB1 reversal. However, we did see adenosine 2A reversal with beta-pinene and geraniol, but not linalool and alpha-humulene. So again, this is complicated. I don't expect everyone to sort of memorize which ones are involved, but just understand that you have the different terpenes are sort of working through different receptor systems in different ways to achieve these behaviors. And again, we might be able to use this. You might be able to dial in the right terpene blend that will synergize in the right ways the pain relief, but not synergize with these other side effects to produce the effect that we want. And a brief note too, I mentioned in the beginning that we use male and female seeding mice for everything. And in general, we did not see sex differences. We just combined the data. That is not true for linalool, for tail flick, and hypolocomotion. So we actually did notice a sex difference here. And so when we have the linalool alone, the tail flick antinociception, there was no sex difference. That's combined. Rimonabant fully reversed in males and females. That was not different. So that's combined. However, we saw a difference in the synergy between WT 55212 and linalool, but that differed between males and females. So first of all, it's good to be a male in this assay. So the males had greater pain relief that lasted for longer. You see this nice strong, synergy or increase of synergy here. However, not so good to be a female if you want to take linalool and relieve your pain. It took longer for the pain relief to come in. The pain relief really wasn't synergized or increased at all. And then again, it's fully gone by 2 hours. So I would term it the females actually had less effective pain relief once they combined cannabinoid and linalool together, but the males they did. So that has potentially important implications, and you might not want to use linalools if you're a female, at least if these results are to be believed. And then lastly, we saw some mechanistic differences in the hypolocomotion between males and females. So the estradiol reversed in females, and the Rimonabant, let's see, the Rimonabant in males. Yeah, the Rimonabant reversed in males. Here, that's significant. And the estradiol reversed in females, suggesting there's a mechanistic sex difference in the hypolocomotion. Adenosine 2A for females and CB1 mediated for males. But these are minor, so these are the only real sex differences we discovered in our studies. All right, so this is our second set of conclusions. So we know that the terpenes induce tetrad behaviors. However, it does, so that's sort of cannabimimetic, hence the title at the start of this talk. However, they have different mechanisms of action that do include the cannabinoid 1 receptor, which is particularly important for antinociception, and synergizes in antinociception with WIN, but differs for the other behaviors. So the adenosine 2A receptor appears to modulate some more of the sedating properties, particularly the catalepsy is very strong. However, there are other systems are involved as well. And in particular, hypothermia, we didn't really get much insight into what mechanisms were being engaged in the hypothermia. There was a minor effect of either antagonist. And we need to look, and there are potential sex differences in linalool as well. So again, overall, we're producing this cannabimimetic suite of behaviors, but they differ between the terpenes and they use different mechanisms of action. And ultimately, we hope to be able to use this to get that right terpene blend. I keep saying this again and again, but I want to hammer this idea home. Use the right terpene cannabinoid blend to give you maximum pain relief while minimizing your side effects. And so again, we come back to our scheme for what we're testing. We have now tested our findings in the mice. And so now we want to go in vitro to support some of the findings that we found in the mouse. And in particular, we're going to focus on the CB1 receptor in an in vitro model to start to try to really to provide some in vitro evidence that the terpenes can engage this receptor, which was suggested by our in vivo studies. And so to do this, we're going to look at ERK map kinase phosphorylation as one signaling output of CB1 receptor activation. So you have your terpene, cannabinoid, whatever comes in, activates the CB1, you get ERK phosphorylation. We measure that. And the way we measured that really depended upon the Azure Sapphire system. So this is where I give my little pitch for the Azure Sapphire. It's a great system. We have this is the scanner. We have the 4 laser version, so it gives you kind of that maximum flexibility, but you can easily accomplish this data with a 2 laser set. And we have 2 main assays that we perform to get data for ERK activation. First of all, we just have a very traditional Western blot. These are some images that we captured using the Azure Sapphire. Very, very clean data, looks very nice, but of course this isn't exactly challenging or novel. This is a Western blot. Everyone can do this. Where I find the Azure system really shines, and particularly this laser scanner system versus an LED illumination system really shines, is the ability to do more complicated experiments, including in-cell Westerns. So you can't do an in-cell Western on a diode type thing. You can't get the right focus. So using this laser system, you can program the depth of the laser, so you can scan the bottom of a microplate, or you can scan the bottom of a slide, or whatever you want to do. But in this case, this is performed in a microplate, and we're able to use a 384 well microplate. So that's 384 data points for one plate's worth of scanning. You culture the cells, treat with drug. Each set of 2 rows is a different drug, and then going from left to right is a dose response curve, and then the green is phospho-ERK. So you have your different drugs. This is a more potent drug. You see stimulation of phospho-ERK. This drug is less potent. On the bottom is vehicle. And so you're able to sort of quickly and effectively and efficiently capture a lot of data on protein biochemistry and activation in this in-cell Western format. I love this assay, and the Azure is the perfect platform to be able to do that. So that sales pitch over. But we really were able to use this equipment to good effect to get our findings. So let's do that. So we have our cannabinoid 1 receptor expressing CHO cells. That's our in vitro model, and we just start by very simply stimulating them with terpene alone, and looking at activation of ERK using the Azure system. Simple Western blots, simple concentration response curves. And we have our Win55212 positive control. And we can see here first that alpha-humulene and beta-pinene, the first 2 terpenes, fully engage the CB1 system. They produce full stimulation of ERK MAP kinase. It's fully activated by 500 micromolar. And we see the same thing with geraniol and linalool, although the doses or the concentrations are shifted. We don't really see any activation until we get to 500 micromolar. And perhaps the stimulation is a little bit less. And now if you're savvy about culture systems and sort of in vitro molecular pharmacology, you might say, hey, wait a minute, 500 micromolar is a really high concentration, right? How do we know that this is anything real at all? How do we know that we're not just, jamming up the system, non-selectively activating, whatever you might say? So there's a few pieces of evidence to suggest that this is real, that this is true engagement of the cannabinoid 1 receptor by the terpenes to produce a true agonist sort of effect. So first of all, by inference, there are other low potency agonists out there that have been shown to be selective that activate in this dose range. So you don't have to have a subnanomolar drug to be a true drug. You can have drugs that are in the micromolar range that are actually engaging these receptors or different receptors, including the cannabinoid receptor type 1. And so just by sort of analogy or inference, this could be legitimate. And second, we're able to fully block this agonist activity. So here you see the agonist activity, the ERK activation. We can fully block that with Rimonabant pretreatment in all cases. So in all cases, selective antagonist treatment was able to fully reverse the stimulation that we saw. And that to me, and including in your Win55212 positive control, right? It acts the same. So to me, this is strong evidence that the terpenes are actually engaging the cannabinoid 1 receptor in a true agonist fashion, because you can reverse it with a selective antagonist. So this is a strong piece of evidence suggesting that the terpenes are engaging the CB1, which supports our in vivo data, if you'll remember, suggesting that the tail flick antinociception was produced by CB1 stimulation. However, we also found in vitro evidence for polypharmacology. So remember, we have this idea from our in vivo studies that the terpenes are engaging multiple receptor systems to produce different effects. And so this came, first of all, when we counter-screened our terpenes in a wild-type CHO. So these are CHO cells not expressing CB1 or not expressing any cannabinoid receptor that we're aware of. Win55212 positive control, 0 stimulation. Interestingly, linalool and geraniol also 0 stimulation. However, alpha-humulene, beta-pinene, and beta-carotene control all produce strong stimulation of ERK, just as strong as it did in the CB1 CHO. And this stimulation was not reversed by cannabinoid receptor antagonist, unlike the previous data set. So previously we saw full reversal. Now we don't see that reversal. This suggests that these terpenes, these 3 terpenes, are engaging some other receptor system present in these cells other than the CB1 receptor, which again fits with this idea that we had from the in vivo studies that these are polypharmacological agents that are engaging multiple receptor systems. Now we don't know exactly what that receptor is yet, But we have some ideas. So adenosine, of course, is one. We're going to chase down adenosine 2A, and we're going to do some screening experiments to identify other receptors. Further supporting this polypharmacology idea, though, is we also tested the terpenes in cannabinoid 2 expressing CHO cells, so cannabinoid receptor type 2. And interestingly, in these cells, we saw full activation by all agonists, by all terpenes. The geraniol is not statistically significant, but I'm pretty sure it's activating there too, because CHOs normally have very low ERK baseline. So we can show now that these are all activating cannabinoid receptor 2, suggesting that these terpenes could have a beneficial effect in an inflammatory type pain, which CB2 has been implicated in. But overall, again, supports this idea that the terpenes are engaging multiple targets, not just CB1 or not just one target. And so this is the conclusion of our in vitro studies. We do have some of our cannabinoids seem to be not selective is not the right word, but seem to sort of engage the cannabinoid 1 and the cannabinoid 2. Well, first of all terpenes engage the cannabinoid 1 and the cannabinoid 2. Some terpenes, alpha-humulene, beta-caryophyllene, and beta-pinene, engage something else that's present in these CHO cells. We don't yet know what that is, but they're engaging CB1, CB2, other receptor systems of some sort to result in ERK MAP kinase stimulation. And at least at the CB1, this can be blocked with a selective antagonist, suggesting that it's a true agonist type effect. We also looked at arrestin and cyclic AMP outcomes, which showed the same thing. I didn't have time to show these today, but we have other cell signaling cascades that are activated as well. And this supports our in vivo studies. And that can be blocked by Rimonabant. Always forget that I have the animations queued up. And then, so that was our in vitro conclusion. The in vivo conclusion, all 4 terpenes are producing analgesic effects that's mediated by the cannabinoid 1 receptor. They're producing hypolocomotion and catalepsy that's produced at least in part or heavily by the adenosine 2A receptor. Hypothermia is produced by some other receptor system, which we really haven't activated yet. But you have this suite of behaviors is being engaged by different receptor systems by the terpenes. And they all synergize with cannabinoid to different extents. They all fully synergize in pain relief, but synergize to different extents in the different side effects, suggesting that you can come up with that one right terpene or that one right terpene blend that will maximize your pain relief and minimize your side effects. And that's really something that we're looking forward to doing and working on now. And also provides at least some conceptual support for the entourage effect hypothesis, which I mentioned in the beginning. And so for future directions, we want to chase down a lot of these areas. In vitro, we want to look at the adenosine 2A receptor since we identified that. Study more terpenes. Of course, we only looked at 4. There are 140 in the plant that we can potentially look at. And maybe identify other receptor targets as well that have been engaged. In vivo, we want to look at, more fully flesh out these relationships, do different doses, create dose responses, determine whether this interaction between terpenes and WIN, particularly in tail flick antinociception, is synergistic or additive, and really get into that. We want to get a little more translational by looking at inhalation, since most people are taking these drugs by the inhalation route. Along those lines, we want to look at THC instead of WIN to come up with more translatable findings. And then we also need to flesh out sort of our therapeutic profile of these drugs, really figure out if they're going to be effective for managing pain. So look at side effects like tolerance, look at different pain outcomes other than simple reflexive tail flick, look at other pain models, in particular neuropathic pain is something that we want to look into. We have some preliminary evidence, which I didn't have time today, which suggests the terpenes may be effective in chemotherapy neuropathy. So there's a lot of different paths to go down looking with these drugs. But they have some promise. And now we're able to show that they have some real pharmacological effects and they may synergize in vivo. So hopefully we can provide some rigor to this whole field and start moving forward. So these are the people that made all this possible. In particular, I'd like to highlight these 3 in bold that were sort of the primary authors and generated all of this data. And really the lead for that is the recently graduated Dr. Justin Levine. He finished his PhD in my lab based on the strength of this project. He graduated last April and is now at Purdue in Val Watt's lab doing a postdoc. Attila and Ryan also contributed very heavily to these studies. And then these are the other members of my lab who maybe did not directly contribute to this study, but are an important part of the environment. And at least for this study, I only have institutional funding to report. We don't yet have any direct funding for this exact project. And so thank you. And I'm going to hand it back off to Justin, who I think wants to say a few words, and then happy to take any questions. Also happy to have you guys contact me separately, right? So if you want to discuss this, if you want to discuss collaboration, if you need tips on how to get the Azure to work for you, whatever, Azure Sapphire to work for you, whatever you want to know, if you want an Incel Western protocol, whatever you need, happy to help. So I'll pass it back off to you, Justin.
All right. So thank you again, John, for sharing this interesting work with us. We are super happy that our Azure imaging system could be a part of your amazing research. So as John has shown, Azure's instrumentation is capable of using digital imaging to analyze receptor activation via Western blots as well as in-cell Westerns. So for any experiments requiring the accurate quantification of fluorescent or chemiluminescent signals, a digital imager or scanner is gold standard. So we offer a wide range of systems for digital imaging, fitting the needs for any application or budget. So with the Azure imaging system, you'll have the ability to perform chemiluminescent Westerns, fluorescent Westerns, gel documentation, and more. So just like John has shown in his research, with the Azure Sapphire, you can image a multitude of sample types with resolution down to 10 microns, as well as have the option of 4 distinct laser channel excitation. So to learn more about our products, reagents, or workflows, you can visit our website at azurebiosystems.com. And yeah, so that just about wraps it up. Thank you, everybody, for taking the time to attend this live webinar today. A follow-up email will be provided within 24 hours in the recorded presentation. I'll also include John's email in case anyone would like to reach out to him directly for any questions. But yeah, so that's going to wrap it up. I'll do the meeting now then.